Hello guys, so today we shall be discussing another important topic, right, which is also called as a sexually transmitted disease, that is Haemophilus ducreyi, right. So we are discussing about Hemophilus ducreyi. So what is this Hemophilus ducreyi, how is it transmitted, and what is the pathogenesis of this? Everything we shall be discussing right now. Now regarding the properties of this bacteria, right, so we shall be starting on with the properties of this bacteria. So can anyone tell the first thing what we have to discuss in case of properties, guys? The thing we have to discuss is whether it is, what shape is it? Is it a coccii shape or is it a bacilli shape? Obviously this is somewhat like a coccii, at the same time like a bacilli. For example, this is the shape of that, this one. So let me draw another picture. You see, this is the shape. So it is somewhat circular, at the same time it is also rod shaped. So it is like a coccii, at the same time it is like a bacilli as well. So I call this as coccobacilli. So this is coccobacilli, right? Second important thing which you have to discuss is that whether this is a gram positive or a gram negative bacteria. Again I am telling you, those bacteria, right, which have got a thick peptidoglycan layer are called as gram positive bacteria, and those bacteria which have got a thin peptidoglycan layer are called as gram negative bacteria. And this bacteria has got a thin peptidoglycan layer. So this bacteria has got a thin peptidoglycan layer. So as a result of this thin peptidoglycan layer, I call this bacteria as gram negative bacteria. What type of bacteria is this, guys? This is gram negative bacteria. Third important thing which we have to discuss is that what type of organism is this? Say this bacteria can live in the presence of oxygen, at the same time in the absence of oxygen, right? So such organisms which can live in the presence and in the absence, they are called as facultative organisms, right? So they are facultative anaerobes. They are called as facultative anaerobes. Fourth important thing is that this bacteria do not have any kind of flagella or cilia. So if it is not having any kind of flagella or cilia, do you think can this bacteria move? No, it cannot move. So this bacteria is called as non-motile bacteria. This is non-motile bacteria. Fifth important thing is that regarding the enzymes, right? So this bacteria has a capacity to release cytochrome oxidase enzyme. So if this bacteria has a capacity to release cytochrome oxidase enzyme, so can I call it as oxidase positive organism? Yes. This is called as oxidase positive organism, right? And the sixth important thing is that this bacteria do not have a capacity to release catalase, so it is called as a catalase negative organism. For example, let me draw the organism here. Let us say this is an organism, right? Now surrounding this organism, guys, all of you should know, surrounding this organism you have got something called, what is this? This is called as a capsule, right? So if this bacteria is having a capsule, can I call it as encapsulated bacteria? Yes. So this bacteria is encapsulated. This is encapsulated bacteria. And second important thing is that from this capsule you have got some things that are coming out like this, right? From this bacteria you have got some things that are coming out like this, some projections. So what are these projections called? Yes, these projections are called as fimbriae. These projections are called as fimbriae. I'll explain you later on what is the important thing about this fimbriae and all. I hope all of you know from the introduction to bacteriology. I have explained you what is the function of fimbriae, what are the function of cilia, flagella, pili and etc. Right? Right. So what is the thing which I wanted to discuss after drawing this picture is that this bacteria releases an enzyme called catalase. For example, right? Obviously this bacteria do not release catalase. Let us say for example this bacteria released catalase, right? So obviously all of you have to know what is the function of catalase, guys. Catalase will convert H2O2. What is H2O2? H2O2 is hydrogen peroxide. So catalase will convert hydrogen peroxide into water plus half mole of oxygen, right? So water and half mole of oxygen, together they form so called as foam. That is the reason why whenever there are any cuts, any superficial wounds, you add hydrogen peroxide. Even during ASDs, I mean the dressings which you do in your clinic, you add hydrogen peroxide and do the dressing, you wash the wound. So you see a foam that is coming out of that, right? So why the foam has to come? Why the foam has to come is that, for example, this is your blood vessel over here, right? Now, in this blood vessel, all of you know that you have got your endothelial cells. So, these are the endothelial cells of your blood vessel. On both the sides, you have got endothelial cells. Let us say that this blood vessel, there is a damage to this blood vessel and there is a rupture of this blood vessel. As a result, the blood is oozing out now. From the ruptured endothelial cell, from the damaged endothelial cell. For example, see, this is the cut endothelial cell, right? This is the cut endothelial cell. From this, an enzyme is released that is called as catalase. And what does catalase do? Catalase will convert hydrogen peroxide to water and oxygen, right? So you will find water in the blood vessel, at the same time you will find oxygen molecules in the blood vessel. Now don't get confused that just now you told me that catalase is released by bacteria and now you are telling me catalase is released by the endothelial cells. Yes, catalase is basically released from the endothelial cells, but catalase is all, but some bacteria have a unique property to release catalase. You know, some bacteria have got some unique property to release catalase. But whatever it is, whatever I'm discussing in the blood vessel is completely different from that bacteria. Forget about that bacteria. Now concentrate on the blood vessel. What is the thing you have to concentrate is that right now in your blood vessel, if you are adding hydrogen peroxide from that broken endothelial cells, catalase is released and that catalase will convert the added hydrogen peroxide into water and oxygen. So you will find many oxygen molecules that are floating in the blood vessel like this. Many oxygen molecules. But okay, fine. So what is the thing you have to know? What is the thing you have to know is that never ever add hydrogen peroxide to deep cut wounds. Why? For example, if there is a cut of brachial artery, ulnar artery, radial artery, or whatever artery it might be, these arteries are pretty big. When you are adding hydrogen peroxide, the oxygen molecules will be formed a lot, right? Many oxygen molecules will be formed within the blood vessel and all these oxygen molecules join together to form a very big air bubble, and this air bubble will cause a specific embolism called as air embolism. For example, you see this big one is called as an air bubble. So these air bubbles here, what are they going to do, guys? They cause air embolism. They cause air embolism. So what do they cause? They cause air embolism. That is why never ever add hydrogen peroxide to deep cut wounds. Okay. But anyways, this bacteria does not release any kind of, let us say this bacteria is not releasing any kind of catalase enzyme. So I call it as catalase negative organism. So for a note, let me take it that this bacteria is catalase negative organism. And finally, the last important thing which I want to highlight here is this bacteria is having a capsule. So I call this as encapsulated bacteria. Encapsulated bacteria. And all of you should know one thing, that I told you Hemophilus ducreyi causes a sexually transmitted disease, right? So it causes a sexually transmitted disease in the sense, how are you telling it causes sexually transmitted disease? How am I telling is that patients who are infected with this Hemophilus ducreyi will have an ulcers, painful ulcers on their body, and these painful ulcers on their body is called as chancroid. Okay, so these painful ulcers on their body are called as chancroid. So what is chancroid? Yes, tell me what is chancroid? Chancroid is a sexually transmitted disease. Sexually transmitted disease. It is a sexually transmitted disease, right? Now, second important thing which you need to know is that, for example, if you wanted to look at this bacteria, if you are so eager to look at this bacteria, so obviously you can look at this bacteria on a special medium. First of all, you have to grow this bacteria. You have to what? Grow this bacteria, in the sense you have to culture this bacteria. And how are you going to culture this bacteria, guys? Yes, how are you going to culture this bacteria? Look here. If this bacteria has to grow, then this bacteria needs a special factor that is called as factor 10. This bacteria needs a special factor called as factor 10. And what is the name of this factor 10, guys? The name of this factor 10 is hemin. Factor 10 is also called as hemin. So if you provide factor 10 to this bacteria, then this bacteria will eat up that factor 10 and grow up very happily. Right? So where will I find this factor 10? Yes, I will find this factor 10 in a special agar, and that agar you call it as a chocolate agar. Right? So you call this agar as chocolate agar. So if I am asking you where I can culture this bacteria, you can tell that this bacteria can be cultured on chocolate agar. Why? Because bacteria needs factor 10, that is hemin, and this hemin is located in this chocolate agar. Right. Next important thing. Let us discuss about the virulence factors of this bacteria. Let us discuss about the virulence factors. Now, regarding the virulence factors, what all things you need to know? First thing I told you that this bacteria is having a capsule around it, right? So can I write that this bacteria is encapsulated? Now you can ask, okay, fine. So if a bacteria is having a capsule, then what's the big deal about that? Capsule is the first virulence factor and the most important virulence factors, guys. Why? Because those bacteria which have got its capsule, when the phagocytes they try to attack this bacteria, this capsule will prevent that phagocytes to attack. This capsule will not let the phagocyte or the macrophage to engulf the bacteria. So can I tell that this capsule is antiphagocytic? Antiphagocytic. So what is the virulence about this? This is an antiphagocytic. So in this way, the bacteria can very freely move in your bloodstream. So because even the macrophages are present near it, they cannot engulf them. Right? Second important virulence factor. Second important virulence factor is that in the previous picture I have shown you that surrounding the bacteria you have got some structures. These are called as fimbriae. Isn't it? You have got some structures called as fimbriae. So the second important structure is fimbriae. And what are these fimbriae and why is it so important? See, basically these fimbriae have got some proteins within them, right? So for example, let us say this is the bacteria over here, right? And let us say this is the capsule over that bacteria, right? And let us also tell that you have got some fimbriae like this. These are the fimbriae. So when you closely examine the fimbriae, within this fimbriae, you have got some minute red color dots which I am drawing, right? So these red color dots are nothing but called as fimbriae proteins. And what are these fimbriae proteins, guys? They are FLP1, next one is FLP2, and third one is FLP3. So these are the basic three types of fimbriae proteins which are located in this fimbriae. But anyways, this is just for an MCQ. But what is the function of fimbriae is that fimbriae are responsible for the attachment to the host cell, right? So attachment to the host cell is also a virulence factor. Third important thing is that surrounding this bacteria, there is also a special structure like this. Look at this now. Surrounding the bacteria, there is a special structure in this way. You see this structure here? So this structure which I'm drawing right now, this is called as lipo-oligosaccharide. This structure is called as lipo-oligosaccharide, that is LOS, right? So what is the function of lipo-oligosaccharide, guys? Basically lipo-oligosaccharide releases toxins, right? But as far as this Hemophilus ducreyi is concerned, this lipo-oligosaccharide, that is LOS, what is its function is, it is responsible for adhesion on the cell. That is, it is responsible for cell adhesion. It is responsible for cell adhesion. So these are the basic introductory parts which we have to discuss. We have discussed about the shape of this bacteria. We have discussed that this bacteria is going to cause chancroid, and I will tell you how chancroid is caused in the pathogenesis. And this is a sexually transmitted disease, and these are the properties which you have to remember. And obviously regarding the virulence factors, so these are the virulence factors basically. Okay. Now coming to the point, what is the pathogenesis? How this is going to be pathogenic to me? What is the pathogenesis of this bacteria? Pathogenesis. So before I explain you the pathogenesis, guys, so let me draw a simple picture. One important thing which you need to keep in mind, is that I told you that this bacteria is a sexually transmitted disease which results in the formation of a painful ulcer called as chancroid. And you know where these chancroids are formed? Chancroids are formed on the genital regions. For example, in the males, they are formed on the penis. For example, in the females, they are formed on the vagina. They are formed on the, in your anatomy you have studied, surrounding the vagina you have got minor lips, right? Labia minora, labia majora. So there these chancroids are formed, right? So let us discuss how are they formed. Let us discuss that. Let me draw a picture. Let me tell you, so this is the male genital organ, that is the penis. This is the male genital organ. And let us say this is the blood vessel. Don't think this is a urethra. The red thing which I'm drawing here is a blood vessel. Okay. And let us also tell that this has got some very beautiful epithelial cells here. Obviously, skin has got epithelial cells, right? So there are epithelial cells on either side. Now what I'm going to do is that I'm going to put up a bacteria over here. So this is the basic representation of bacteria which I'll be giving. And how this bacteria enters into this epithelial cells, then enters into, first how this enters into the epithelial cells and how it enters into the blood vessel. First thing you need to keep in mind is that this bacteria enters into your body through the open cut surfaces. For example, if there is an open cut surface here, from this open cut surface the bacteria enters inside. Okay. Even in the mucosa also the same thing. Even in the genital mucosa also the same thing. If there is any open cut injury of the genital mucosa, from these open cut surfaces, skin surfaces, the bacteria very easily enters inside. Okay. So let me put up a picture here. Now what I'm going to draw is, I'm going to draw epithelial cells on one side, blood vessel on the other side. What I'm trying to tell you is, I'm going to draw this part, epithelial cells and the blood vessels, right? So let me do my picture over here, right? So this is your blood vessel. Let us say this is your blood vessel over here, right? Now surrounding this blood vessel, I told you, you have got epithelial cells, right? If I look closely enough, look here now, blood vessel in the center, on the lateral sides you have got the skin. Blood vessel in the center, on the right side I have drawn the skin. Now surrounding the skin, what should I draw, guys? Obviously I have to draw the epithelial cells. So these are the epithelial cells like this. These are the epithelial cells. Now this entire layer which you can see here, this entire layer which I can see here, right? So this is called as dermis. This is called dermis. And this layer here is called as epidermis. Epidermis. Nothing more I have discussed here. I have just zoomed the picture inside. And second important thing which you need to know is that lining your blood vessels, you have got endothelial cells, isn't it? Lining your blood vessels, you have got your endothelial cell. Let us say this is one endothelial cell, right? This is the next endothelial cell. This is the third endothelial cell, fourth, fifth endothelial cell. The same on either side. I have just enlarged one endothelial cell to show what's happening in the blood vessel. Okay, but don't worry, I'll still prolong this picture if I have to discuss more pathogenesis here. Fine. So I have the bacteria here. Okay, let me create something. There is an open cut surface. Let me put up the margins. You see, there is an open cut surface of the penis. Now from this open cut surface, this bacteria has got its red carpet, and very easily, very nicely, this bacteria, as if it is walking in a red carpet, it is very easily entering into your epithelial cells. Sorry, the endothelial cells. So right now, this is a bacteria, right? This bacteria is located within your endothelial cells. Now the moment the bacteria enters into the endothelial blood vessels, this bacteria will stimulate the endothelial cells so much that the endothelial cells release two important things. What are those two important things? It will release interleukin 6, that is one cytokine, and it will also release another cytokine, that is interleukin 8. Okay, fine. So first it is going to release interleukin 6. Now what is this interleukin 6 going to do, guys? Look here. This interleukin 6, what will it do is that this interleukin 6 will call all the CD4 T cells. It will call all the CD4 T cells. So what will happen is all the different types of CD4 T cells, they come here. So many CD4 T cells are here now. CD4 T cells. Now what this interleukin 6 will tell is that it will tell all the CD4 T cells to go to a specific place. You know what is the specific place? Where did the infection start? Where did the entry start? The entry has started near the epidermis and the dermis region, right? So this interleukin 6 will order all these CD4 T cells to go to that epidermis and dermis and see what bacteria is there, and if there is any bacteria, just kill down the bacteria. So obviously all of you know I've just represented one bacteria, but in fact there are multiple bacteria over here. There are multiple bacteria. So give me a minute to represent this. There are multiple bacteria over here. So now you tell me where CD4 cells and all of this go. Obviously the CD4 T cells, they come here. The CD4 is here now. The CD4 T cell. So instead of writing CD4 T cells, I'll just write it as T cell. Okay. So now all the T cells are here. Okay. So these are the multiple T cells. Now, you know what these T cells will do? There is bacteria there. T cells are also there. So there will be a fight between these T cells and the bacteria. So in the process of fighting, where are they fighting? Outside? No, they are fighting in our own tissues. So in the process of this fighting, the dermis and the epidermis will be damaged, right? And that will create a chancroid, an ulcer. So how is that happening? Not only T cells alone will cause a chancroid, but there are also some other mechanisms. But the first mechanism which you need to know is interleukin 6 will call for CD4 T cells, and T cells will go there to the dermis and the epidermis region where the bacteria is there, and it will start a fight. There will be an immune fight towards the bacteria. Second thing, I already told you that this endothelial cell has got activated right now, right? So it even releases not only interleukin 6, but it also releases interleukin 8. Can anyone tell what is the importance of interleukin 8? Interleukin 8 is a chemoattractant molecule. Interleukin 8, this is a it is a chemoattractant molecule. Chemoattractant molecule in the sense it attracts, what cells does it attract? It will attract your this cell, what is this cell called as? This is called as a macrophage. Yes, it will attract your macrophage, and it will even attract your, let us say, neutrophils. It will even attract your eosinophils. It will even attract your basophils. Now tell me, neutrophils, eosinophils and basophils together, they are called as polymorphonuclear cells. They are called polymorphonuclear cells. And the other one is your macrophage. So this interleukin 8 is a chemoattractant molecule. It is attracting two important things. One is your polymorphonuclear cells, that is PMN cells, and the other one is your macrophages. So what will happen right now? What will happen right now is that you will see macrophages here in the blood vessel. At the same time, you will see what? Tell me, you will see, obviously you will see eosinophils in the blood vessel, you will see basophils in the blood vessel. Along with eosinophils and basophils, you will even see a specific cell that you call it as a neutrophil. I'm not drawing the nucleus of that because by now you should know how does a neutrophil look, right? How many lobed is neutrophil? So a neutrophil comes here. Okay, fine. So what is this macrophage and neutrophil going to do in the blood vessel? What is it going to do is that within this macrophage and within this neutrophil, a small reaction is going to happen. Look what that reaction is. The reaction is that, so let us see what is happening in this macrophage. In this macrophage, you have got an oxygen molecule. And this oxygen will be converted into O2 minus, and this O2 minus, guys, this O2 minus, you call it as a superoxide ion. This O2 minus, you call it as a superoxide ion. Again, I am telling you, these superoxides, peroxides, right? These superoxides or hydroxide radicals, whatever are there, these are radical molecules. Radical molecules are very, very dangerous to your body. These are all called as free radicals, like superoxide, right? OH dot, which is also called as hydroxyl radical. So all these radical molecules are too much toxic to you, that these radicals are so powerful that they can even cause lysis of your blood vessels. They can even break down your blood vessels. And even if even bacteria, any kind of bacteria in your body is afraid of these radicals, because radicals are so powerful that they can even break down the bacteria also. So what these macrophages and neutrophils will do is that these macrophages and neutrophils will convert oxygen into superoxide ion. And these superoxide ions go all the way back and kill down the bacteria. But that thing is not going to happen here. The moment superoxide ions are released, bacteria is so aware that, look here what is happening, it is very interesting. Bacteria is so aware that this bacteria releases an enzyme. You know what that enzyme is? That enzyme is copper, zinc, superoxide dimutase. What is that enzyme? That is copper zinc superoxide dimutase. Copper zinc superoxide dimutase. And this copper zinc superoxide dimutase will not make this reaction to happen. Will never make this reaction to happen. So superoxide ions are not formed in this condition. When they are not formed, they stay as oxygen molecules, and hence the bacteria can live very freely. Now the bacteria thinks, I am absolutely free now, I can divide, and now bacteria starts to divide. And bacteria, once it settles in the endothelial cell, it releases, it releases all these enzymes like superoxide dimutase and all of this, right? And make sure that no one is touching them, right? Now the bacteria is free. It can do whatever it wants. Now slowly the bacteria thinks, I'll start releasing toxins and start damaging this guy, this blood vessel, his penis, little by little, Now what is happening? What is happening is, look here. Let me elongate this blood vessel. This is the blood vessel over here, and these are your endothelial cells. These are your endothelial cells, right? These are your endothelial cells, respective endothelial cells, right? Now look what is going to happen. So this bacteria right now is in your endothelial cell, right? So let me draw this bacteria. So this is your bacteria, and this bacteria is right now in your endothelial cell. And now look here very carefully. This bacteria within this endothelial cell releases two different types of toxins. Okay, what is that one toxin called as? That is cytolethal distending toxin. So it releases a toxin that is called as cytolethal distending toxin, which is also called as HDCT. HDCTD. Okay, so it is abbreviated in the name HDCTD. HDCTD stands for, yes guys, HDCTD stands for cytolethal distending toxin. Very important, keep this toxin in mind. So this releases a cytolethal distending toxin. You know what this toxin is gonna do? This cytolethal distending toxin, look here now, every cell, if it has to divide, what process will it undergo? Mitosis and meiosis, obviously, right? So only when there is a cell cycle, only in that, because with the help of that cell cycle, the chromosomes also they divide, and the cell also divides, right? So for example, this endothelial cell, within this endothelial cell, there is such cell cycle happening. So in the cell cycle, you know, you have got G1 phase, G2 phase, M phase, S phase, and all of these phases, right? So what this cytolethal distending toxin will do is that it will all the way go and look at that cell cycle. And in that cell cycle, there is a specific stage called as G2 stage. There is a specific stage called as G2 stage. It will inhibit this G2 stage. It will inhibit this G2 stage. When it will inhibit this G2 stage, so there will be no cell division. No cell division. When the cell cannot divide, the cell death will happen. Right? The dead cell will transform itself into a necrotic cell. So finally, a necrotic cell is formed. Okay, so this entire endothelial cell, I'm showing it to you in the next picture, look there, the entire endothelial cell, it becomes necrotized like this. So there is necrotization of the endothelial cell. The same concept which I have explained you, look here now again very carefully. So these are the cells of the penis. Now what will happen is that the same concept is going to apply even in the epithelial cells of the penis. The same thing, within the epithelial cells of the penis, what is happening is this bacteria is going to release cytolethal distending toxin, and this cytolethal distending toxin will cause necrotization of your complete cell. So that is the reason why you even find necrotization of some of the cells of your penis. So your penis turns black, right? Your penis turns black. Not the black which you think, but this is something little more black, you know, right? So this is the concept over here. There is necrotization, right? So two things are happening here, guys, to the penis. First thing is the T cells came there, they caused some sort of damage. And overall, the toxin is released, and even they are also causing necrosis and some sort of damage. Now look step by step how penis is getting affected, right, by all these immune attacks and all. On the other hand, you know what will happen? On the other hand, this bacteria on the other hand is going to release another toxin, and that toxin is named as hemolysin. That toxin is named as hemolysin. You know what does hemolysin do? Within your blood vessel, you know, you have got T lymphocytes, you have got B lymphocytes, and what else? You have got macrophages. T lymphocytes, B lymphocytes and macrophages. So this hemolysin is going to break down your T lymphocytes, B lymphocytes and macrophages. When it is going to break down your T, B and macrophages, your immune activity, your immune capacity is lost. Your immune cells are lost. When your immune cells are lost, can you fight with the organism now? You cannot fight with the organism, right? So overall guys, overall, two things you have to know, that what is a toxin that is releasing, that is hemolysin and cytolethal distending toxin. What is the enzyme that is releasing? Copper zinc superoxide dismutase. Next important thing is, what are the cytokines is it releasing? Interleukin 6, interleukin 8 and its functions. Okay, so these things are very important to remember. Fine. So when I look at the picture of the penis, how does it look? This is the penis, and if you see, you will find damage to the penis at multiple levels. Multiple damages. You know how the damage is happening right now? Because of the toxins that are released, because of the interleukins which are released. So you will see a multiple ulcers, you will see a multiple ulcers on the penis like this. And these ulcers are surrounded by a gray border, right? How are these ulcers looking, guys? These ulcers are looking like this. Gray yellow base with irregular borders. Now don't think the borders are fine. Why did you tell it as irregular? Okay, let me draw it as irregular now. So these are irregular borders. Okay, so gray yellow base with an irregular border is a characteristic representation of the ulcer. And this ulcer, guys, this ulcer which I have mentioned right now, this is called as your genital ulcer or chancroid. So from today onwards, if someone asks you, can you please depict chancroid? Yes, chancroids are mainly formed on the open cut surfaces of your skin as well as the mucosal surfaces. Second thing is, chancroids have got a grey yellow base with irregular borders. Chancroid is basically an ulcer which is painful, which is a painful ulcer, right? So you can ask me one question that you told me previously. You can ask me one thing that you told me that because of this necrotization, because of the cytotoxins that are released, because of these interleukins, you told me there will be a great damage to the epithelial cells of the penis, that is in the epidermis and the dermis, and that is resulting in chancroid. Yes, exactly, that's what I told you. But if you very closely look, guys, if you very closely look, very simple thing, if you very closely look, what is the first thing that is happening? Let me rub out one thing. Let me rub out one chancroid here. The first thing that is happening is, you will first see red color papules like this. First you will see a red color papules on the penis. These are called as erythematous papules. What are these called? These are called erythematous papules. And these papules are so close to each other, but by chance what will happen is that all these papules will join together. They will join together into a very big single papule that is called as pustule. If all these papules join together, you call it as a pustule. Okay. Next, if this pustule ruptures, okay, let me show it to you. If all these papules join together, you're gonna find a pustule like this. Okay, the second picture here which I am showing to you right now is your pustule. And if this pustule grows big and finally bursts out, how does the picture look now? So this is the bursted pustule, and this pustule is surrounded by some yellow color thing with some irregular borders. So pustule bursts out and the resultant is chancroid. So now you understood what are the stages of formation of chancroid. The first thing is erythematous papules. Erythematous papules fuse together to form pustule. Pustule ruptures to form a chancroid. And chancroid is a painful ulcer, very, very painful ulcer. Now in these patients, one very important thing you need to keep in mind is that these patients will not have any kind of systemic symptoms. Systemic symptoms is, for example, if a man is having herpes, he will have fever, he will have nausea, he will have some other symptoms, right? Along with the herpes rash over his body. Along with that, he will be having fever, headache, nausea, vomiting, and all of these things. So these things are called as systemic symptoms. But this patient will not have fever, he will not have cough, not cough, he will not have fever, he will not have any kind of sepsis, he will not have any kind of hypotension, he will not have any kind of headache, vomiting, no systemic symptoms. Only localized symptoms, and those localized symptoms are located on the genital tract. The patient is coming to you, you are a doctor in the clinic, and he will just complain that, doctor, nowadays I find some painful rashes on my penis. Once have a look. Now if the doctor is so interested over to look his penis, he will look that there are some painful ulcers with some irregular borders that are formed, that is called chancroid. And patient will tell that, I don't have any other complaints, doctor, I just have only that complaint. Okay, so one very important thing which you need to know is that this patient has got no systemic symptoms. And you know what the good news is? Good news is that all these chancroids, they heal within a week. They heal within a week. Right. You get a doubt. Sir, I have studied about Treponema pallidum in my class, and I have also studied about herpes virus in my class. Even in Treponema pallidum and herpes virus, we have got some ulcers in the genital region. Now, how are you going to tell, if I look at an ulcer, what should I think? Should I think that it is Hemophilus ducreyi, or should I think that it is herpes, or should I think it is Treponema pallidum? Keep in mind one thing, guys, that in Treponema pallidum, yes, I agree with you that in Treponema pallidum, you have got ulcers. But these ulcers are what? Non-painful. These ulcers are non-painful. These non-painful ulcers located on the genital tract, these are called as canker. They are chancroids. This is canker. Second thing, you also told me about the herpes, right? You reminded me about the herpes. So even in herpes you find the ulcers? Yes, even in herpes I find the ulcers. But the differentiating factor is, in herpes, yes, I agree that there will be painful ulcers like Hemophilus ducreyi. There will be painful ulcers. Along with the painful ulcers, patient will have systemic symptoms. But what about our bacteria which we discussed, the Hemophilus one? In that there are no systemic symptoms. Okay, so this is the thing which you need to keep in mind, right? So in the females, I told you, right? In the males on the penis, you will find these rashes. I'll show you the picture, but I don't have the picture of these chancroids which are formed on the female genital tract. But let me, draw it and make it clear to you. So let me draw this thing. Very rough picture. So this is the vagina of the female. And what are these things, guys, which I'm drawing, right? The wavy thing which you can see there, that is labia minora. Surrounding the labia minora, the fleshy outline which you can see is called as labia majora, right? So these are the legs over here, right? These are the legs, and I'll show you where these are formed. So you make this female to sleep in this position, right? So she's sleeping in this position. Now she's sleeping in this position. Now you see on the labia minora, On the labia majora, you will find multiple ulcers. On the labia minora, you might find a single ulcer, but on labia majora, you will find multiple ulcers. Right? So all these ulcers, these are called as what? Chancroids. Okay, so this is labia minora and that is labia majora. So this is the place where you will find the ulcers. And second important thing is that in the females, one very interesting thing you will find that you will have a swollen lymph node on one side only, not two sides. So this is called as unilateral lymphadenopathy. You will find unilateral lymphadenopathy. Now, when it comes to the treatment part, what is the basic treatment which you give to the patients? When it comes to the treatment part, what is the basic treatment which you prescribe for your patients? The basic treatment which you prescribe for your patients is azithromycin or ceftriaxone. Azithromycin or ceftriaxone. Right, so let me show you the pictures of the genital ulcers. So in the first picture you can see that there are multiple erythematous papules, right? So all these are erythematous papules. All these are erythematous papules. So all these erythematous papules, they join together to form a single one. This is called as pustule. This is called pustule. And the pustule bursts out to form an ulcer, which you can see here. You see an ulcer here? And this ulcer is surrounded by an irregular margins which is yellowish in color. So this is what is called as your chancroid. This is your chancroid. So this is the end of the lecture. Thank you.